Hello everyone, I'm HEERANYAA NADARAJA. Today I'm going to perform on SDS page in lab stimulation. What is SDS page? SDS page refers as sodium dodecyl sulfate polyacrylamide gel electrophoresis. This is a straightforward and low-cost method for separating proteins from complex mixture. SDS page gels will be used to analyze the yeast protein extracts that prepared in previous lab. Usually, two gels will be created to visualize all of the proteins in the extracts. One gel will be stained with the simply blue. The second gel will be used for Western plots, which will look for MET and LACZ fusion protein extracts. Let's stimulate. Level 3. Protein gel electrophoresis is a laboratory technique that separates protein by the mass. This technique is similar to running an agarose gel to separate and visualize different molecules of the DNA. After the electrophoresis, the visible protein will be typically visualized by staining the gel with cormacy. And in this case, we use the Western plotting, which is a common technique which allows for the specific detection of the just one protein among many with the help of specific antibody. Western plotting is a routine technique to test the protein expression of specific gene in the cell. Now, let's talk about the materials that we're going to use for this experiment. There are several materials like reagents, micropipetting equipment, empty tubes, electrophoresis equipment, and other equipments. First, 4x SDS page buffer solution. This buffer solution usually used to prepare protein samples for denaturing gel electrophoresis with triacetate gels. It needs to be diluted 4x to have the correct final concentration in the sample. And here we have a wild type cell culture, which is a tube containing a culture of a wild type unedited macrophage cells. Pancreas cell culture is a tube containing a culture of the pancreas cell. Edit for cell culture, it's a CRISPR edited macrophage cells, which is known as edit 4. Edit 7, same like edit 4. Protein leather solution, it's a protein leather containing pre-stained proteins of the following sizes such as 10, 15, 25, 35, 45 and so on. And one times running buffer, which is a salt containing buffer to cover gel and transmit the charge through the protein gel, allowing the electrophoresis to occur. And micropipetting equipment, we have P2 micropipette, P20 micropipette, P200 micropipette, and P1000 micropipette. And here we have the micropipette tip box. Empty tubes, we have wild type cell tubes, we have pancreas tube, we have edit 4 tube, and we also have edit 7 tube. Polyacrylamide gel, which is a gel consisting of a molecular self polyacrylamide that separate SDS coated protein by the mass. Protein gel electrophoresis box, which is the box holds the gel upright and filled with the 1x running buffer. Power supply, which is supplies the current. Power supply leads, which helps to flow the electricity through the gel. And here we have a microcentrifuge, to have a high speed to the collected liquid at the bottom of the tube for the easy transfer. Biohazardous waste, which is help us to eject the micropipette tips, and heat box, which help us to incubate the sample at a constant temperature over a long period of the time. Next is procedure. Let's read through the procedure. Here we have 6 steps. First, we have to collect the cells. Second, we have to add the sample buffer. Third, we have to denature the protein. Fourth, centrifuge the sample. Fifth, load the gel. And sixth, conduct the protein electrophoresis. Let's start stimulation.